Welcome to the University of California, San Francisco Sports Medicine Podcast, featuring Dr. Nirav Pandya, Dr. Brian Feeley, and Dr. Drew Lansdowne, discussing hot topics in sports medicine and society. We hope you enjoy our podcast and look forward to hearing from you.
Welcome, everyone, to our UCSF Sports Medicine Podcast, 68 Weeks, with myself, Dr. Nirav Pandya, and Dr. Brian Feeley. Today, we have the pleasure of having Dr. Rando on. He is a world-renowned neurologist and stem cell researcher who currently is leading stem cell research over at UCLA. So thank you for joining us today, Dr. Rando. We appreciate you being on. My first question for you is, tell us a little bit more about what got you excited about science and how that got you to where you are today.
Well, first, thank you, Nirav, and thank you, Brian, for having me on. It's a pleasure to be here. What first got me excited about science, and I think I was always excited about science as a kid, and I think the big transition for me was deciding, as I was leaving college, how much to focus on research and how much to focus on clinical medicine. And I was applying to medical degrees. I had done research as an undergraduate, and I've always made it my goal in life to have undergraduates in my lab as a way of giving them that opportunity to explore a career path they might not otherwise even think about. And it was critical for me, and I knew I loved it. So I ended up deciding to go into an MD PhD program. So I actually didn't make a decision. I just decided I'd do both and maybe make a decision later. So that's, that was how I ended up continuing from some very simple things done as an undergraduate through where I am now. But as time went on and I completed my clinical training in medical school and my residency, I felt the draw of lab research increasingly. And so throughout my career, I combined clinical medicine and research, but research just took a much bigger part of my academic efforts.
So I wanted to start a little bit basic and talk a little bit about what stem cells are and why they're important when we study aging. So at the most basic, straightforward level, when you describe a stem cell, what is it and why do you think they're so important when we study how we get old and why we get old?
So, good questions. So stem cells come in different flavors. I'll start with kind of the type of stem cell that we study. And the stem cell that we study the most is the stem cell in muscle, the tissue that makes you move. And why are stem cells important? Stem cells are important because in muscle, the cells that make your body move are called myofibers. They're very large. They contract, and that's what makes your muscles contract. Whereas the stem cells are these small, almost spore-like dormant cells that sit around and don't do anything, thank goodness. They just sit there. However, if muscle's injured, and if you've ever walked downhill, done some hiking downhill, and 2 or 3 days later, you find your muscles are incredibly sore, that's because the muscles have been damaged. And what's happening during that process is these rare stem cells that are scattered throughout this dense tissue, they begin to wake up and they make more of themselves, and then they begin to make new mature muscle. And so in that sense, stem cells through many, many tissues in the body, but not all, are present to maintain tissues and repair them after injury. So why is that important as we age? Well, as we found out, and this has been studied long before we started studying this, well known that with age, this process of stem cell function declines. And we get tissues that repair less well, less effectively, less rapidly. I mean, if for all of you who have kids or have had kids, you see that when they cut their skin, they have Wolverine-like healing. They just, they heal and there's no scar. It's just like beautiful. And the older we get, you notice that it heals more slowly and less effectively. And that's true in every tissue, in skin, in bone, in muscle. So what we study is, why is that? Why is it that the cells that are there to repair tissue do less well as we age? And is there anything we can do about it? Can we induce these old stem cells, or whatever's wrong with them, to repair tissues like young stem cells? And that's been a major focus of our lab.
And kind of continuing on with that point, where do you see that transition of quote unquote people getting old in terms of their muscle? When does that process kind of, in the timeline, your 20s, your 30s, when does muscle and all these changes start occurring?
Well, so, if you look at muscle function, depends on what sport you look at, and you guys know this better than I do, but, marathon times peak, I think, in the 30s or early 40s, whereas sprinting time peaks much earlier. So it sort of depends on what you want to measure as what you want to say. What's the peak and when do you start to decline? But generally, one peaks in terms of performance in most sports around the 20s, some plateau, and then you start to decline. But there's no magic age or even decade that you could say you can't get better, you can't improve. I mean, so there's a lot of flexibility and modulatability in terms of muscle function. But I would say that post puberty, early 20s is when muscle function, strength, and endurance and are peaking, and then they stay high and then they decline after that. In terms of
sorry, I'm going to butt in. It's definitely before 48 because I do not recover the way I used to even 3 years ago.
Yeah, I can attest to that too. I'm a runner, and I did. Well, I was a late life runner, so I don't have anything to compare to in my 20s or 30s. I started running in my 40s. My times in the marathon went down, but that was just from practice. That wasn't my muscles getting better. But without a doubt, every few years, it's those post-run injuries that are taking a little long to recover. And I think you really notice that in one's 40s, 50s, 60s, and beyond.
So you had a really cool paper that came out relatively recently, and it talks a little bit about the differences between our young and old stem cells. What I thought was really cool, and I may have gotten this wrong, was that when we get old, we still have stem cells. They still seem to act like a stem cell, but they may not have the same capabilities. So what have you seen when you study the difference between young and old muscle stem cells, and what does that mean in terms of our ability to recover and regenerate?
So that's an interesting question with an interesting answer. And let me wind back the clock a bit. So we did some experiments back in the early 2000s, which really opened up a field in regenerative biology and aging. It's kind of an intersection of those two. And the experiments that we did were experiments called parabiosis. And that's where two animals, in this case mice, are surgically connected. And they develop a single shared circulatory system. So the blood of one animal begins to circulate through the tissues of the other animal, and vice versa. It goes both ways. What was interesting is that when we connected a young mouse to an old mouse, so now you have young blood components circling through the old mouse tissue and old blood circling through the young mouse tissue. What we found was that the old mouse that was experiencing this exposure to young blood, the tissues in that mouse, and in particular, we looked at muscle stem cells, began to behave like young stem cells. So that meant that there's something in the environment, there's something circulating in the blood or some things that are influencing these quote old cells to become young again. And by the way, it works both ways. The young cells exposed to the old blood begin to work less well. So the whole idea was this aging axis that we've been looking at looks like it might be modulatable, malleable, that you can actually, in a sense, and we've gone on to study this, program old cells to become younger and young cells to become older. So back to your question, Brian, if with age, the reason why the muscle repairs less well is likely a combination of aging of the muscle cell itself, plus the aging environment. The circulating factors that are coming from old blood that are having a kind of suppressive effect or negative effect on all tissue regeneration. So in that sense, to make old muscle repair better, you could either target the factors that are suppressing it, or you could just target the cell itself to see if you can restore youthfulness to an older cell.
Tell us a little bit more about what's in the blood itself, particularly for younger patients, kind of in this process. Like, what are we seeing, particularly for that younger age group?
So if you look at blood and you just ask what's in it, it's a very, very complicated tissue, right? In addition to the cells that are in the blood, the white blood cells, the macrophages, the red blood cells, there's all the proteins and lipids and small molecules. And we and others have been looking at the composition of blood and its change with age. And it's so many things change. It's hard to pull it down to specific patterns, except, for example, old blood looks more inflammatory. There are more inflammatory proteins in old blood. And I would say that has to be, at least the lion's share of and there are a lot of factors that are like this, that are in old blood that would, when delivered to a young animal, suppress that activity. There's kind of a pro-inflammatory environment with age. Young, it's harder. There are more pro-growth, as you might expect. A young boy or girl, they're growing rapidly. You want factors that are promoting growth. So there are a lot of more growth factors and factors that will promote tissue expansion and cell proliferation. So to generalize, and there are many other things that are changing, to generalize, it might be going from a pro-growth environment to an anti-growth environment. In fact, you could argue that there might be an evolutionary benefit for once we reach reproductive age, we don't want to grow anymore. We've reached our size, and what we don't want to grow are things that we don't want to grow, like cancer. So, and that's when you start to see, over the life course of a person, mutations develop in tissues. There might be an advantage to suppressing growth in cells that you don't want to grow. So that's hand-waving evolutionary argument, but it fits with the idea that this goes from a growth phase to a non-growth phase and more of an inflammatory phase.
All right. So if I have this correct, we have an 11-year-old, Stu, who's going to be about 6'5". I can this evening go get his blood, transfer it to me, and then I'll be young, and then I'll keep growing. Correct?
That's correct. You'll be 15 again.
Awesome.
So we've done these kinds of experiments where we've given, so not through parabiosis, but actually through blood transfusion, given young blood to old animals and old blood to young animals. And what you see is effects of the blood on the animal. They don't, they don't get younger, but aspects of their tissue improve to be like a younger, in this case, animal or person. We haven't done this in people, but so the idea is that there are programmable aspects of aging that you can restore. And kind of the Gundogan experiment, if you take this to logical extreme, there would be rejuvenation of the individual if, but what we don't know is what the downsides, and almost certainly there would be, of giving lots of young blood to older people. I mean, one in particular is what I just mentioned. If someone has some nascent cancer that is being controlled by their own immune system, and you suddenly give them a series of factors that makes that cancer grow, bad news. So, so this is going to be, I think this is going to be an interesting balance in finding pro-growth factors and anti-growth factors that will define how we age, how cells age, and what kind of therapeutics we can envision could be feasible.
Yeah, and I think it's interesting when we look at kind of the clinical aspects of things. We've concentrated a lot on, how do we extend our lifespan in terms of preventing disease. But a lot of this research seems to, kind of think of this concept of health span. How do you improve, a non-pathologic state? Is this what people refer to when they're thinking of kind of science and medicine as health span as opposed to lifespan?
Yeah, that's exactly right. I mean, so in the aging field broadly, when it was observed, back in the 1930s, that you could make rats live longer by feeding them less calories. Simple intervention, caloric restriction. They live longer, and actually they live healthier, but they live longer. And so a lot of the focus as model organisms to study aging were developed, whether that's Drosophila, the fruit fly, or C. elegans, the worm. The easy measures, how long do they live? So the literature on interventions like drugs and diet and things like that, over the decades, especially in the 20th century, the focus was on lifespan. How long did they live? But when you start talking about human lifespan, what we don't want is, we don't want humans living longer sick. Our goal is not to extend the number of years that people live after they've already lost function, cognitive function, motor function. The goal really is to have them not even live longer, live the same amount of time, but with less disability. And so that's where the notion of health span, not lifespan, came into kind of common parlance, and has really taken over lifespan. Now, that having said that, lifespan is easy to measure. Is the fly dead or alive? Is the worm dead? Health span is a lot harder, as you know, in humans, to determine sort of the frailty of a person. You've got to measure a lot of things, and you've got to take a lot of time. So health span conceptually is what we focus on. Practically, it's just a more difficult measure.
Okay, so you've gotten older. You've already mentioned that now somehow you run marathons, and you study aging. You're probably one of the 5 foremost researchers in the world studying aging. So what do you specifically do to maintain your health span without making us all do marathons?
So, what I do, I'll tell you, start with what I don't do. So I don't take supplements that are, touted as promoting health span. I'd say I do a few things. So first of all, I keep a relatively paleo-ish diet, meaning I try to eat things that I think our bodies were designed to eat. Okay, so that tends to be, relatively low carbs, fruits and vegetables, some meat, but pretty much low carbs and low salt, and so I just try to keep a diet that feels consistent with evolutionary selected pathways. I run. I mean, again, I run marathons for fun. I never started doing it for health, but I think that the benefits of many of the drugs that are being tested for lifespan are really intersecting with this balance between diet, exercise, and weight. And I think that if I could recommend, this sounds like what your mother would recommend, eat less and get more exercise. It didn't take all those years of medical degrees and research to come up with that, but that's what I've come up with, is that for the most part, the things that we study and we're really focusing on, when I say we, I mean the field, it comes down so often to this balance between what you put in and what you do. So the diet, and it's linked to obesity, or if it's caloric restriction, it's linked to starvation. You can't go too far. So I think of everything in life in this world as being on an inverted bell-shaped curve. Too much exercise, too little exercise, too much food, too little food. You want to be reaching that that maximum. And if you're there, adding more supplements or adding more drugs, I would argue, is unlikely to have a major effect. And I think when we see benefits in health span in animals, say in laboratory mice, I think a major reason why they're living longer in response to this drug or that drug or this treatment is that they live in a world where they eat all the time. We feed them so-called ad libitum. They have food 24 hours a day. And as they get older, they get fat. And if you really want to do a test in a laboratory animal in terms of whether a treatment, let's say a drug, makes them live better or healthier, you first have to eliminate that issue of the excess food, excess eating, and obesity. If you can keep the animal in a more natural state, and in the wild, animals are generally hungry. They're not fed. And so that's a more natural state. And then say, will this drug help them live longer and healthier? I think it has much more meaning.
And one last question before we wrap up, kind of in line with that. Obviously, my practice is more kind of pediatric patients, and we always tell our patients the decisions you make now will impact you as an adult. When you look at it from a basic science level, do you see things over a prolonged time when patients are younger that can then impact their ability to deal with aging and tissue degeneration when they're older?
We've never studied this in humans in terms of any of this kind of regenerative biology, but I think there's a wealth of evidence that even in utero, what children are exposed to can have a profound impact on their susceptibility to disease. Now, we don't understand this well, and this falls into the realm of epigenetics. But I think it's unequivocal that what we do as our children go through, development in utero, postnatal development, childhood development, adolescence, what they do has the potential to impact their susceptibility to disease many, many years later. Now, we don't understand that well, but I think that that's present, and that's why I think it's very important to think about their health not only as their child health, but also leading to their adult health.
Well, thank you. We could talk for hours about this. There's so much at the tip of the iceberg with this, but thank you for joining us today. We really appreciate you taking the time, and good luck with all the amazing research that you're doing. We just, as surgeons, I feel, well, Brian obviously does research. This is just well beyond anything that I'm capable of. So thank you for doing all the work that you do.
Well, thank you. It's, I love talking about this stuff. So thanks for having me on.